Question number 151. In gel electrophoresis, the separated band of DNA containing the gene of interest is cut and extracted from the gel piece. This step is known as, and the options are denaturation, spooling, DNA isolation, and elution. And the correct answer to this question is option number 4, elution. Elution is a step preceded by gel electrophoresis, in which the separated band of DNA containing the gene of interest is cut and extracted from the gel piece. Let's discuss rest of the options. The very first option given is denaturation, and this is an incorrect option. Denaturation is a separation of DNA strands at high temperature. The next option is spooling, and this is an incorrect option. Spooling is the process by which precipitated DNA can be removed from suspension. And the last option is DNA isolation, and this is an incorrect option. DNA isolation is a process which involves separation of total DNA from the cell culture. So the correct answer to this question is option number 4. Let's proceed to the next question. Question number 152. While isolation of DNA from tomato, RNA can be removed by the treatment with, whereas proteins can be removed by treatment with. Select the option which fills the blanks correctly. And the options are, the first option says lysozyme and cellulase. The next option is ribonuclease and protease. Third option is chitinase and lipase. Fourth option, protease and deoxyribonuclease. And we know enzymes are named after the substrates they act upon. So if RNA or ribonucleic acid is a substrate, the enzyme would be ribonuclease. And if proteins are substrates, then enzyme would be protease. Whereas rest of the enzymes, such as in first option, lysozyme and cellulase, this is an incorrect option because lysozyme and cellulase act on bacterial and plant cells respectively. The next option, ribonuclease and protease, is a correct option and is a correct answer to our question. Chitinase and lipase is an incorrect option. Chitinase acts on fungal cells, whereas lipase acts on fats. The last option, protease and deoxyribonuclease, is an incorrect option because protease acts on protein, whereas deoxyribonuclease acts on DNA. So the correct answer to this question is option number 2. Let's proceed to the next question. Question number 153. Select the incorrect statement with respect to gel electrophoresis. And the very first statement given is, DNA fragments can be separated by forcing them to move towards the cathode under an electric field through a medium. And this is an incorrect statement and is a correct answer to our question. As you can see here, DNA is loaded on the wells towards cathode, which is a negative electrode. And then this DNA fragment moves towards the positive electrode known as anode. So this is an incorrect statement because DNA fragments can be separated by forcing them to move towards anode and not towards cathode under electric field through medium. Medium is what? Agarose, which is extracted from seaweeds. So the next statement, commonly used matrix for gel electrophoresis of DNA is agarose extracted from seaweeds, is a correct statement, so cannot be a correct answer to our question. The next statement is, DNA fragments separate according to their size through sieving effect provided by the agarose gel, as it is also a correct statement, so this cannot be a correct answer to our question. As you can see here, under electric field, these DNA molecules get separated according to their size, and the larger the size, the more they are towards cathode, and the smaller fragments move farther towards anode, as DNA is negatively charged, so it will move towards positive electrode. The next statement is, we can't see pure stained DNA fragments even in visible light, and as it is also a correct statement, so this cannot be a correct answer to our question because pure DNA is colorless and is not visible to naked eye. In order to see it, we need to stain it with Yttrium bromide and need to visualize it under UV rays. So the correct answer to this question is option number 1. Let's proceed to the next question. Question number 154. Intercalating dye Yttrium bromide stacks between base pairs of DNA and stains it. Upon exposure to UV light, DNA appears, and the options are dull blue, bright green, bright orange and yellow. And the correct answer to this question is bright orange, option number 3. As you can see here in this gel picture, we can see bright orange colored bands. These are the DNA bands of DNA fragments. These are bright orange DNA bands when stained with ETBr, ethidium bromide, upon exposure to UV light. Whereas rest of the options, dull blue, bright green and yellow, are the incorrect options. So the correct answer is option number 3. Let's proceed to the next question. Question number 155. Select the first restriction endonuclease isolated and characterized from Haemophilus influenzae. And the options are EcoR1, Hind1, Hay1 and Hind2. And the correct answer to this question is option number 4, Hind2. Hind2 is the first type 2 restriction endonuclease isolated and characterized from Haemophilus influenzae. And let's discuss rest of the options. The very first option given is EcoR1, and this is an incorrect option. EcoR1 is a restriction endonuclease which is isolated and characterized from Escherichia coli RY13. The next option is Hay1, and this is an incorrect option. It is a restriction endonuclease which is isolated from Haemophilus aegyptius. So, EcoR1 isolated from Escherichia coli RY13, Hay1 from Haemophilus aegyptius, and Hind1 from Haemophilus influenzae, but it is not a type 2 restriction endonuclease and is isolated later to Hind2. So Hind1 is an incorrect option, and the correct answer to this question is option number 4. Let's proceed to the next question.